All right, so let's really talk about the skeletal muscle now. Now, when we look at the skeletal muscle, we call them fibers. Why? Well, it's because of their elongated cylindrical-like shape. And their length ranges from less than a millimeter to over 12 centimeters in humans anyway. So it's really the longest cell in the human body. It's pretty impressive for it to be like, 12 centimeters. That's pretty cool. Now, these are multinucleated with peripherally located nuclei, and they have striations with them. Right? And if I drew that up here in my striations, and just to kind of, I guess, maybe recap quickly, I do have a little drawing here. Remember, we have our muscle, right? Our whole muscle belly, and then we cut that, and we have all these fascicles, right? What are fascicles? That's the bundle of muscle fibers. And inside the fascicle, we have our individual muscle fibers, right? So muscle, fascicle, muscle fiber. And then we want to zoom in on this because you can see the striations, right? You zoom in on this and we can see what's going on a little bit more inside of that muscle fiber. Well, what is it, right? We see that each of these sarcomeres, like what we see right here, these are the smallest contractile unit of the muscle fiber. That's what gives, as I said, the striated appearance, and that's how the muscle fibers are organized. And each sarcomere contracts independently of one another, and they're still functionally coordinated within the motor units, right? But they contract independently from one another. Now, let's talk about that muscle cell some more, or a myofiber. Now, again, it's enclosed by the sarcolemma, which is the specialized plasma membrane, and it has a lot of these. What I drew here, these red things, these are called T-tubules. Okay, I was going to label that here. T-tubules, okay, for transverse tubules because they go through that entire thing. And they're basically invaginations that are continuous with the sarcolemma. So literally you have your sarcolemma here, and they come down. Like, here's a good example. They literally come down into the sarcolemma. So I probably could have done the same color, but I want to really have these T-tubules stick out. They're part of that plasma membrane of the sarcolemma, and they're literally, like I said, these invaginations with that, and they contain the sarcoplasmic reticulum, or the SR, which is the specialized portion of that cell that contains lots of calcium, and essentially it's a storage. And it releases calcium during contraction. And there's also the terminal cisternae, or these lateral sacs, okay, right here. And that's where it's going to store all of this calcium. And when we have like a T-tubule down right here, and the lateral sacs that come right by it, we can call that a triad, all right? And that's going to be important when we release the calcium from the sarcoplasmic reticulum. Now, another thing about these muscle fibers is that they have multiple nuclei, right? Or they're myonuclei, and they have mitochondria. Okay, so you have like a nuclei over here. They have mitochondria right here. So it's the same cell, so it has a lot of all the organelles that are in there. And the mitochondria, of course, are really important. And there's going to be this subsarcolemmal mitochondria, and that's involved in signaling and ATP support. So it's like right underneath the plasma membrane. It's sitting right underneath there. And then you have your intermyofibular ATP, and that's more important to supply muscles for contraction. And then again, you have all the other organelles like your Golgi apparatus, your ribosomes, and so on, all the other cellular components that are important for your cells, peroxisomes, and so on. So now let's go and zoom in on this a little bit more, and we're going to look at the myofibrils and the sarcomeres and this whole contractile organization, right? So you have the myofibril itself, and this has the myofibrils, and they're like cylindrical contractile structures running longitudinally, okay? Like they run longitudinally with, I guess we would say like here, like they run longitudinally with the muscle. Within the fiber. And they're composed of these repeating sarcomeres, which is the smallest functional contractile unit of the muscle fiber. Now, these sarcomeres contain many different proteins for them to function. And the primary ones are things like myosin, actin, troponin, and tropomyosin. We're going to talk about these in more detail, but it's really important you know all of these. So let's talk about the sarcomere more specifically. Now again, sarcomeres are going to be arranged in a series within the myofibrils, producing these striated appearances. That's why the skeletal muscle has these striations, because of the sarcomeres. And this individual contractile unit, they're about 2.2 micrometers in length, and it's about 4,500 sarcomeres per centimeter of the muscle length. All right. Now, if we dive into the sarcomere more, we have the different filaments, the thin filaments and thick filaments. Now, the thick filaments, so I'm going to take here, I'm going to zoom in on this, and we're going to have this specific myofilament. And we have our thick filament, which is here, and our thin filament, which is here, okay? Now, this here is primarily myosin, and this one here is primarily our actin. And there's different proteins that are going to be with this. Now, with the thick filament, like I said, that's going to be your myosin, and it has your globular heads right here. That's going to be attaching to the actin when we come to cross-bridge formation. And then we also have the thick filament has a titin in it. And here we have this titin molecule that goes from what we call the Z disc, and it goes all the way to this line, which is called the M line. And that's really what helps stabilize the thick filament in position within the sarcomere. Okay, so now the thin filament, that's primarily composed of F-actin. Okay, these two guys here, that's F-actin, or it's an actin filament, so we call it F-actin, which is essentially polymers of G protein, right? So if you just have, for example, each one of these is actin, right? And if you put a whole bunch of them together, then you get a filament. So we call it F-actin. And it's going to be like 300 of these G-actins in order to make a filament. So we call that F-actin. And then you have 2 of these actin filaments that are surrounded or that are spiraled together to make that thin filament. All right. Now, along with this F-actin and these that you also have various associated proteins, such as the nebulin, and that's this green protein right here. And this is an important protein because it helps to stabilize a thin filament, and it kind of acts as a ruler to help when your cells are making the filament, they got to know how long to make it. So they go along until it reaches the end of that nebulin, and now it knows, okay, that's as far as we go, right? And it's also attached there to the Z disc. Okay, so that's what nebulin. It's a pretty big protein, really. Now, if we go and look more at the sarcomere structure and the banding, okay, so we think a lot in terms of banding of how the sarcomere is structured and then how it functions. Because remember, structure dictates function. So if we understand the structure well, we can really understand the function of everything. So when it comes to the sarcomere, we have these, first the Z disc, all right? Now, that's this part right here, which is called the Z disc. You're going to want to know all of these parts to really understand the sarcomere. Now, the Z disc is this boundary, and it anchors the thin filaments, and it's right in the center of that is what we're going to call the I band. All right. So you have the I band that's going to be over here, and this we could actually draw out another. So it'd be another sarcomere over here until you get more that's going. But here you have what we call an I band. Okay, this is your I band, and then as I said, the Z discs goes right in the center of that I band. All right, and when you're thinking about, I know your text on the PowerPoint slides that we prepared has some really good images, so you can look at those a little bit better. But it's also important to look at how these are arranged. Now, when we look at the I band, also during contraction, what happens is the Z lines, they're moving closer together. So that as the sarcomeres are starting to shorten, the Z bands are going together, all right? But these Z discs, they really are what anchors like the actin, and they have the titin and everything connected to it. So Z-discs will be very important as we continue going forward with this. Now, another important one is called the A-band. Now, the A-band is going to be the length of the myosin filament, okay? So we can go like this, all right? And this here is called the A-band. Now, this is important because, so A-band stands for anisotropic. So basically just when it was discovered and you know, how it polarizes and light, it's going to be darker. It's not as uniform. Iso means like uniform, and so it's not uniform, it's anisotropic. So that's why it's the A-band. That's why it's called that. But it's really just the length of the myosin. And it can include regions of overlap within the thin filaments, except within the H zone. So as you can see, like here's the actin filament here, and there's some crossover, right? And then there's crossover right here. But the A-band is just the myosin. And I mentioned the H zone. The H zone is pretty much right here, okay? This is going to be what we call the H zone. So during muscle contraction, you can imagine that as your myosin heads pull the sarcomere, the Z disc together, right? Then your I-band is going to get shorter, and your H zone is going to essentially disappear until your actins are kind of hitting each other, right? But your A-band always stays the same length, right? Because again, the A-band is just the length of the myosin. All right, so that's going to be important to understand that. Now, the other part is there is the M-line protein. Yeah, I want to make sure I mention that. This M-line protein, or just the M-line, it's made of M-line proteins, but M, it's the midline of the sarcomere. It's the center of the H zone, and it's a structural anchoring region for the thick filaments, and it aligns the point for all the sarcomeres. All right, this is important for just the entire structure of the sarcomere, but we'll talk a little bit more about that also. And this is going to be important for the whole cross-bridge cycling and the ATP, right? So if you remember from your earlier classes, but we'll talk about it more here, the ATP binding to the myosin causes the detachment of the actin. That's important to remember. So ATP itself, it's used not to get a power stroke, but actually just so that the myosin heads detach from the actin in the first place. And the ATP hydrolysis to ADP and the inorganic phosphate that's bound, that's what cocks the myosin head. And then during that, so if you have the myosin like this, right? If you can imagine, I can do this, huh? So the myosin is attached to the actin. ATP binds it, and now it can relax. And then it hydrolyzes, and it cocks the myosin head back. All right? Now it's able to bind, it binds the actin again if there's a calcium signal. We'll talk about that later. But if calcium is present, then the actin binding site is available. Myosin's going to bind to there, and now it's going to remove that removal of the inorganic phosphate. That release drives that power stroke. And then later, ADP, which is still stuck there, it just, it goes away, it releases, and that completes the cycle. And then it's going to do the same thing again. If calcium is still present and everything, well, first off, ATP, which should always be there because you have your mitochondria, ATP is going to come, it's going to bind, and it's going to let go of the actin. And then if there's no calcium, then even once it hydrolyzes the ATP, it can't bind anymore, right? Because it's being blocked by tropomyosin. We'll talk about more. But it's important to understand the binding of myosin to the myosin heads with the ATP and that cycle, all right? Now, when it comes to the sarcomere, we also want to understand this length tension relationship between with the muscle fibers, because that's going to produce the maximal force when the sarcomeres are at a slight stretch. All right. Now this is, in other words, you need to have some overlap, right? And this is primarily based on single fiber studies, but it really holds true with whole muscle as well. And it's this force optimization occurs because the actin filament ends align optimally with the myosin heads, right? So you get the maximum number of cross bridges that are formed. Because if you think about it, if you're like outstretched all the way here, so you're only getting like one myosin head with the actin, you're not going to be able to contract very much, right? But if you have an optimal level, then you can contract a lot more. And that's why a lot of people are familiar with your strongest, like if you're curling weight, for example, you're going to be the strongest range of motion, like at that 90, well, it's not quite 90, but around that angle, you have the strongest range of motion. And part of that is due to an optimal cross bridge formation, right? That's the length-tension relationship that you want to understand. And then, of course, if you have total, contraction, well, then you can't do anything because you're already fully contracted, and you don't have as much force in that area either. So that's an important area just for application to understand a little bit. Now we want to go through some more of these proteins, and then a lot of this is about proteins and things before we start getting more into the mechanisms of how things are happening. So if we look more at these proteins and how they can be organized, so we can organize them through the sarcomeric or endosarcomeric, meaning within the sarcomere, or there's also extrasarcomeric or exosarcomeric proteins, which means they're outside of the sarcomere. So still inside the muscle cell, but are they within the sarcomere or are they outside the exosarcomeric? And they have lots of functions, right? Like the contractility, there's regulatory, there's the cytoskeleton, there's the intermediate, supportive roles it has, signaling. They're involved in so many different things for the contraction, the architecture, and of course the force transmission. And that's really what we want to emphasize here. So let's talk a little bit more about these sarcomeric or endosarcomeric proteins, right? So again, the myofibrillar contractile protein, that is myosin, okay? That's this big daddy here. That's the myosin protein. And this is this is what we just call the thick filament, really. And then we also have the sarcomeric alpha actin, and this is the contractile protein of the thin filament. And notice that this is, I just said alpha actin, I meant F actin, but this is different from the beta and gamma actin, all right? But you also have, so you have this actin right here, this is F actin. You also have some other actins that are important for the overall structure of the muscle fiber as well. The beta and the gamma are more of the cytoskeletal actin, all right? And then you also have the myofibrillar and regulatory proteins, tropomyosin, we kind of just mentioned, and then the troponin complex, which is a big one. And the whole cytoskeletal and structural roles as well we need to understand, because these maintain the three-dimensional lattice of the structure of the myofibrils. And that's what transmits the force internally and toward the cell periphery. So if we look at the endosarcomeric structural proteins, right? So the cytoskeletal proteins, we have titin, also called connectin. All right. Now this is this big protein here. I can just label that. Right here, this black protein. Okay, I'll maybe use a black pen. This molecule, titin, also called connectin. Now this is a huge protein. It's actually the biggest or largest protein of the human body. It's like 27,000 amino acids long, and it's like 3,000 kilodaltons. And essentially, it connects the thick filament to the Z disc, and it extends all the way to the M line, as you can see, in this drawing here. And it's going to be important because it's basically acts as this spring where it helps the muscles to relax, or when they are relaxed, it pushes the sarcomeres back, and it's kind of elastic in the I band but more stiff in the A band. And again, it contributes to that passive force. And it's possibly damaged during like eccentric production. It's during eccentric contraction, it could be damaged, and so it's like a Z-line streaming is what it's called. Now, the other kind of analogous protein is nebulin. Now, this one lies parallel to the thin filament. That's this guy here. All right. We just mentioned this one earlier, but this is nebulin. And as I said, this one acts, like I said, like a molecular ruler for the actin filament for its length. It also stabilizes the filament and anchors the actin to the Z disc. And it may also actually modulate the actin and myosin interaction a little bit and the ATPase activity at rest. And now when we look at the Z line, this is made up of proteins. So there's Z line associated proteins, right? You have actin to the Z disc, right? That it anchors the sarcomere, the sarcomeric actin to the Z disc. It also anchors the titin, which spans the Z disc and goes towards the M line, and it supports the lattice structure, right? And it's going to be cross-linked with cytoskeleton proteins as well. So, for example, desmin, that's going to be a big one. I'll show you here in a little bit where desmin comes into play at. And then you have the M line, right, or this M region. And the M comes from the German word Mittel, which sounds like middle, because it is. And that's where it is right in the middle of the myosin thick filament. And you have major proteins like myosin, for example, and other M-line proteins with it. And this cross-links the thick filaments and anchors the thin, and it anchors titin at the center of the sarcomere. And also, I want to include this M-line also contains this other enzyme called CK, or creatine kinase, right? That's going to really help to buffer your ATP, because if you're doing all this contraction, you need a lot of ATP, and that's going to help to buffer some of that. So it's a scaffolding protein. It's not a scaffolding protein, but it's more of a ATP buffering protein, but it's right there on the M line, so you want to make sure you have that. So then there's a number of other, but there are other scaffolding proteins, but like the myosin, the M protein, and other ones are important to know. Okay, and then the thin filament, okay, the thin filament associated proteins. This is going to be an important for the regulatory aspects of the whole cross-bridge formation. So you have tropomyosin. Okay, now tropomyosin, I don't have it drawn, but basically tropomyosin is going to be between the helical grooves of the F actin. So here's the F actin right here, and I can grab like over here, and it's going to kind of lay over right on the actin. And it's not super long. I mean, each molecule spans about 7 G actins in 7 G actin units. And then the other one is this troponin complex. And this is going to be like, also like right here. Again, there's some pictures in the slides that you guys have you can find to really help you understand this. But this is the entire troponin complex, and there's different areas, right? There's troponin C, which binds to the calcium, troponin T, which binds to tropomyosin, and troponin I, which stands for inhibitory, but that's what binds to the actin. And it binds to actually the same place of actin where the myosin heads want to bind. So because the troponin I is bound to that area, it's one of the reasons why myosin can't bind, and that's why it's inhibitory, right? So we look at actin as the thin filament. It's the other contractile protein, right? Now, there's about 6 actin filaments that surround each myosin filament, right? And then each actin filament is surrounded by 3 myosin filaments. In other words, you have your actin, but over here, okay, there's another myosin right here, okay? And then say 3-dimensionally over here, over there, there's another myosin, right? So each actin is going to be surrounded by 3 of these myosins. And then every myosin will be surrounded by about 6 of those actin filaments, all right? Now, when we do some comparative looking at the actin-myosin arrangement, this is going to help us understand better things about the force production and the contraction velocity. the ATP turnover rate. Those all come in part of this arrangement, right? Okay, now let's switching gears a little bit to go to extrasarcomeric proteins. These ones are really important for the force transmission, and they're important for the general organization overall, and they can be either intracellular and membranous or membrane associated, and they essentially link the myofibrils to each other, to other proteins, and they localize primarily at the sarcolemma. All right, so we want to talk more about some of these proteins, and I'll show you some drawing here. Okay, one of those proteins is really important is desmin, and this is an intermediate filament protein. So you have like this desmin protein it's basically it's going to combine with a bunch of other ones and make this filament of desmin, right? And it basically encircles the Z disc and it links to adjacent myofibrils laterally. Because again, we have this happening, but it's happening within this muscle fiber. It's a whole bunch of them. And so desmin is essentially going to link all of those Z discs together. So those are the adjacent myofibrils that are laterally linked together. And that maintains the longitudinal alignment of the sarcomeres. And the desmin also, this is intermediates, it has intermediate filaments that connect to the Z discs, to the intracellular cytoskeletal and the perinuclear and the mitochondrial and other organelles networks. And it does that through a linker protein such as plectin, okay, and it also couples other Z discs to the sarcolemma through the plectin-costamere interaction. In fact, I want to show you that. I tried to show you this a little bit here with this arrangement. So again, you have your filaments and your Z discs that we're talking about here. So this is your whole like Z disc, and then we can put some of these proteins that we're talking about. Okay, so this one right here, this protein, we can call desmin. Okay, and like I said, desmin, it can connect to, say, the mitochondria, to the nucleus, to other organelles, but it does that through other proteins like plectin. In fact, I'll just label this one plectin. Okay. And then this is going to be linked further to costamere structures. Now, the costameres are the proteins that link. They basically link all the Z discs to the sarcolemma and into the basement membrane. All right, but the desmin, that's essential for the mechanical integrity and the myogenesis of the muscle cell itself. And it links to the cytoskeletal beta and gamma actin as well, and it forms this intracellular cortical cytoskeletal network, right? So that's the actin that is binding to the desmin, and often through plectin, intermediates, and the filament structure, finally to the costameres. And then more about the costameres. Again, these are like membrane-associated proteins, and they're basically muscle-specific. They also contain these muscle-specific local adhesion-like complexes that are located at the sarcolemma and aligned with the Z discs. And they anchor the intracellular cytoskeleton to the extracellular matrix and finally to the basement membrane. And that is what's really going to help to enable the lateral force transmission. So if we look at the costameric protein complex, right, there's going to be two major complexes that I want you to look at. Number one is the integrin-associated protein complex and the dystrophin-associated protein complex, right? And I wrote these ones over here. So we can say this one here, we can say is the integrin. So the integrin-associated complex, and we'll say this is the dystrophin associated complex. That was with DC, not direct current. But this is the dystrophin-associated complex. And this is just, again, take a look at some of the slides online so you get a better image of this. But both of these bind the cytoskeletal actin inside of the sarcolemma, and they bind the extracellular matrix components outside of the sarcolemma. All right. Now, if we look at the integrin-associated protein complex specifically, now this is, as you can see, it's a there's different isoforms of it. There's multiple of them, but it's a transmembrane receptor that binds the laminin and it binds the laminin in the basement membrane, and it's also associated with various signaling proteins like FAK, the focal adhesion kinase, and other mechanotransduction molecules as well. And then the other one, the dystrophin-associated protein complex, you have the important protein dystrophin. Actually, I want to write that. This one right here, dystrophin. Okay, that's a very important protein, and you can see how it anchors the cytoskeleton actin to the glycoprotein complex, right? And then there's other components like there's beta-dystroglycan and alpha-dystroglycan and myosin and laminin 2 and other associated proteins, right? There's a whole bunch of them. So these are critical for force transmission. And you can imagine if you are missing one of these proteins, like let's say you're missing a desmin protein or you're missing the dystrophin protein, right? What's going to happen? How are you going to get that lateral force transmission, right? What's going to happen? You can still get some longitudinal force that's going on when you're contracting and making your muscles, but you're not going to get that lateral force transmission, which is really important because you're missing the dystrophin protein. So there's various diseases such as Duchenne muscular dystrophy where you're lacking that dystrophin protein, or so you're lacking it maybe from like a nonsense or a missense codon, where in this case you could have a mutation in the genes so that you have a substitution of an amino acid so that the structure is a little bit different. So it doesn't have the same strength or function. It's still trying to, but that's why there's different types of muscular dystrophies, right? But this is why it's important to understand muscle, understand all these proteins, why we go into them, because clinically it really matters. And we really, in this class, really focus on this lateral force transmission. And so I want you to understand how this force goes, starting from the very inside and go outside. So we can start here, like at the sarcomere, and we can think about that you get the longitudinal force, right? So like pulling on a rope, right? So here you get longitudinal force, that's going directly. But then you also have that lateral force that's going out. And this is what you understand is that lateral force is being transmitted from the sarcomeres to the Z discs. And then that goes to the desmin, all right? The desmin proteins and those intermediate filament networks. And then that goes into the cytoskeletal actin network. And now we reach the costomeres, okay? And from the costomeres, we're going to have your integrin and your dystrophin-associated complexes, and then finally to the sarcolemma. And then from the sarcolemma, we go to the basement membrane. And that includes your laminin and, of course, your type 4 collagen, and then finally to the endomysium, and then to the paramysium, and then finally to the epimysium, and then finally to the tendon or the aponeurosis, and then finally to the bone. So you can go through all of these layers, starting from desmin, the smallest protein, well, not the smallest, and really, I guess, starting even from the sarcomere, but desmin is linking all of this whole lateral force chain. You want to understand this entire lateral force chain all the way to the epimysium and onto the tendon and onto the bone. And that's going to be really important to understand all of that structure and that architecture. And now what we're going to do next is we're going to go into what's causing the muscles to contract. We'll go through the cross-bridge cycling. We'll actually go through the depolarizations of the neuron, the alpha motor neuron, organization of the nervous system itself, and go all the way to movement. But now that you understand the architecture, you understand the structure, you can now understand the function.